Hello. This discussion is titled, What if Prions are everywhere? When I first took a good look at CoV-2 and compared it to the first zoonotic coronaviruses, based on the rearrangement and mutations of the end terminals, I almost immediately thought this might be the next prion generator. It was a very good educated guess based on my sleepless years as a multidisciplinary molecular biologist. However, I have now seen enough evidence to consider it not just an idea or a hypothesis, but a theory. A prion disease occurs when a misfolded protein interacts with other like proteins, misfolding them, and so on. And those misfold because the infectious protein is more rigid and like attracts like. So, the misfolding goes viral, so to speak. It is commonly believed to be a brain disease, but prion diseases may very well be everywhere. Perhaps they are believed to be exclusively diseases of the brain because the few prion diseases that have been properly characterized only exist in the brain. Maybe people believe that prions need the protection of the blood-brain barrier so as not to be destroyed by our immune system, though it has been demonstrated that prions can exist in the blood. And it has also been demonstrated that SARS-CoV-2 can invade the brain. I've been suggesting for a while that viruses and bacteria may be causing many of our poorly characterized unknown diseases, which may be most of them, due to prion formation. Thanks to the innovative work by Dr. Prokop and his team, and his admirable open source work on the Viral Integrated Structural Evolution Dynamic Database. This is abbreviated as VISTEDD, VISTED. That's a lot easier. I and all of us have access to incredibly valuable data. Jeremy Prokop's group implemented a sequence-to-structure-to-function analysis to understand SARS-CoV-2 proteins, developing an in-depth understanding of protein conservation, structure, and molecular dynamics for each protein involved in both the structural features of the virus and the function of the virus as it enters your cell and disintegrates into a rebirth of hell. So, through this database, I have seen further evidence that SARS-CoV-2 may be generating their own prions. The nucleocapsid protein, which contains the viral RNA and, of course, has an RNA binding domain. Keep that in mind. The RNA binding domain is a very important thing that I will be discussing further in this podcast. According to Prokop and the VISTED data, the nucleocapsid proteins may also be responsible for potentially hijacking 10% of our translation. Translation is the production of proteins by ribosomes based on the genetic code of the messenger RNA that it is reading. It also has the ability to inhibit what is called nonsense-mediated decay. That's referring to a nonsense mutation. These happen all the time. This is part of the immune system inside each of our cells. There are constantly RNAs coming through with many types of mutations, as well as viral RNAs that don't belong there. Our cells sense that these transcripts don't belong there and has them degraded. SARS-CoV-2 inhibits this process. Not only does this allow the viral genome to be copied and translated into proteins unhindered, it also allows the expression of our own suppressed genes by screwing up our proofreading defenses. For example, skin cells have all the genetic information needed to be bone, blood, brain, you name it. This is why each cell needs to keep most of its genes quiet. We also have ancient genes that are never used anymore. If you like TED Talks, you should look up this guy who is gradually turning chickens back into dinosaurs by using genes that have been silenced long ago. That's some cool stuff. Maybe a little bit unethical. Anyway, as with all life, we function off of checks and balances. For example, if something like a virus manages to get past the nonsense-mediated decay defense, our cells will use a backup mechanism where the mutant protein is recognized and ubiquitinated. Ubiquitin is a ubiquitous protein that is attached to mutated proteins, among other things, so that they can be brought to the lysosomes. It tells our cells to bring this mischievous protein to the lysosome because it's basically a big sack of acid that degrades the protein. SARS-CoV-2 also has a protein that will take the ubiquitin right back off, allowing for dominant negative results. A dominant negative protein is one that is generated by a mutated gene, but gets translated into a protein anyway and remains in the body to cause havoc. An example of this would be the disease brittle bones. Darwin Prokop, who is not the same Prokop as Jeremy Prokop, who I was speaking of earlier, is just a coincidence. Darwin Prokop discovered the dominant negative mutation that in one strand of our collagen triple helix. This mutation causes the triple helix to unravel, and as a result, your bones break very easily, which is why the disease was called brittle bones. Darwin Prokop coined the scientific name for this disease, osteogenesis imperfecta. Though it is not a prion, technically, this is an example of how prions can be formed through parasitic activity. It is the nature of proteins to copy one another in a form follows function mode of operation. Like attracts like is a common theme in the way we function. A misfolded protein could easily interact with another protein, changing its conformation and setting off a chain reaction. Another example, also outside the brain, starts in the intestine. Dr. Tuchel's research, I hope I'm pronouncing his name right, revealed that bacteria that cause gastrointestinal illness, Salmonella in particular, often produce a mutant protein called curly amyloid. Remember, Alzheimer's is caused by the buildup of amyloid plaques in the brain. Reactive arthritis affects approximately 5% of people infected with bacteria that causes gastrointestinal illness. This next part sounds all too familiar to me. There are no treatments and little is known about how or why it develops. Dr. Tuchel also explained that if the curly amyloid escapes the intestinal tract and enters the blood, an autoimmune reaction occurs. This is what causes reactive arthritis. And even worse, the presence of curly amyloid-induced autoantibody and joint inflammation affects the fluids surrounding the joints and fucking up bone reabsorption. So, your bones break down and is a hallmark of reactive arthritis. This same phenomenon also is linked to neurodegeneration. It's funny how me and so many others have fibromyalgia, yet, as far as I'm concerned, it's a name for symptoms, but not a real disease. Not until it is characterized. This is a message for all of you other doctors out there. If you don't know what someone has, don't fucking tell them they have something made up. As a well-seasoned doctor of biomedical science, I personally believe that we should know what something is before we diagnose people with what it isn't. And what about mutations that doctors don't normally look for, if they look for mutations? Do you know how mind-boggling complex our signal transduction mechanisms are just in one cell? Do you know how many enzymes in the Golgi apparatus there are that could have genetic or other mutations, and how important these enzymes are for proper folding and glycosylation of nearly every protein we generate, or parasites generate for that matter? If positive-negative mutations like the one that causes osteogenesis imperfecta can get by all of our checks and balances, why not proteins with a perfect genetic sequence that get misfolded by other proteins in the Golgi apparatus, or bad pH regulation, bad glycosylation, improper phosphorylation, methylation, acetylation, you name it? What if a protein takes the wrong highway and doesn't make it to the lysosome, where it can be sliced up right and proper? It has been shown that neurodegenerative diseases like Lou Gehrig's disease can be linked to dietary exposure and non-genomic amino acids. Also, viruses like CoV-2 have pseudonauts in their genome, which have a vast array of fascinating uses. One involves skipping the stop codon and continuing translation of what we call run-on sequences, which go far into what should be untranslated regions. And this generates derelicts and troublemakers that were never meant to be. Viruses also tend to bring their own non-structural machinery. Many say viruses don't have their own replicative machinery, but they do. Anyway, they can incorporate non-genomic amino acids into their transcripts. Non-genomic means not what our bodies use. Our bodies don't even produce all of our own amino acids. We have to get them from food. What if bacteria in the meat you consumed 5 years ago contained non-genomic amino acids, as many viruses and bacteria do? What if depression, anxiety, hard times change our diets and we can't rely on food to supply the amino acids our bodies can't produce? So our own machinery makes up for it with a substitution. What if we can't afford fresh food, so we take more risks with what we eat? What if doctors can't locate genetic signs of a disease, but assume it's genetic because it seems to run in the family? By the way, families tend to be around each other for significant chunks of their lives. We often adopt the same eating habits along with the way we handle food. Even if they move away, they are often still culturally and behaviorally connected. Look and you will see that most of our diseases are guessed at. Look at fibromyalgia. How come I'm just now getting it after living around others that have it? How come we have so many diseases that are diagnosed based on a general combination of symptoms, even though the supposed disease was given a name, yet hasn't been identified or properly characterized? We get second opinions because we can't decide which doctor is more wrong than the other. Maybe we think prions can only happen in the brain because that's where the specific protein defined as the initiator of misfolding and prion generation exists, as if that is a vital component and the only one. Many believe Alzheimer's is not a prion disease simply because it lacks the PRP protein and accumulates beta amyloid plaques on its own, as if their machinery were malfunctioning, their translational associated machinery. It's almost like something that might happen as you age. By the way, PRP is what most scientists like to call the prion protein. So, maybe this lack of creativity exists because too many scientists are looking for their keys at night beneath that proverbial street light because it's easier to see under the light, even though they obviously lost their keys somewhere off in the dark. What if doctors looking for their keys under that blinding streetlight won't even entertain the idea that most of our unidentified, uncharacterized, and unnoticed diseases are caused by things as minuscule as seasonal flu, the common cold, food poisoning from years ago, hard times, closed minds? Another study found that many of our most basic and essential cellular processes that we've depended on for millions of years are guided by the actions of our most ubiquitous endogenous proteins that maintain our cytoskeleton, regulating nearly all signal transduction pathways, protein trafficking, cell division, etc. They're doing so via prion-like behavior. In the journal Cell, Wang et al. Published an article called A Molecular Grammar Governing the Driving Force for Phase Separation of Prion-like RNA Binding Proteins. It's a mouthful. It was a very good article. Remember, it was the protein terminal ends, specifically of the spike protein and the nucleocapsid with the RNA binding domain, that brought to my attention how easily SARS-CoV-2 can generate prions. Well, our cells are not just sacs of fluid with a nucleus. There is a skeleton that segregates, traffics, and facilitates very specific permutations of cell signaling pathways. It is a dynamic fluid-like skeleton in which the terminal ends of these integral proteins determine phase transition based on their amino acid composition. The skeleton, among other intracellular components, must constantly rearrange by hardening parts and softening others. This is phase transition. It is mainly governed by interactions among tyrosine residues on the prion-like domains, as they call them, and arginine residues on the RNA binding domain. The saturation concentration of phase separation are inversely proportional to the ratio of arginine and tyrosine residues. The biomolecular condensates can resemble liquids, gels, or solids. I'd like to mention one thing I noticed that was unique about CoV-2. It was that mutations at the terminal ends, one in which there was an addition of several arginines flanked by serines. CoV-2 is unique among SARS viruses in that furin, a protease that cuts at serine residues, cleaves SARS-CoV-2 but not SARS-CoV-1 in order to activate it. Another protease called trypsin cleaves CoV-2 4 times more efficiently than CoV-1. Notably, it has been found that aberrant phase transitions are associated with the onset of age-related neurodegenerative diseases. There's much more that I would like to say about this article, but I'm busy. So let me just point out that prion behavior is not just parasitic, it is endogenous. But this paper also gives hope. By focusing on the composition of the terminal ends of proteins that seem to have prion-like behavior, most of our unidentified, unnoticed, and uncharacterized diseases may suddenly be in plain sight and easily dealt with, or at least understood. Such an enormous set of novel data points could change the face of biomedical science. What I am talking about here isn't just the rambling of an inspired dilettante. It is an observation that has likely been noticed by many accomplished multidisciplinary scientists like myself. This is an invitation to join in on a new understanding of how medicine should be approached. I believe that changing our minds should be the prime directive of all scientists. Improving medicine is a moral imperative. I see no reason why prion diseases wouldn't exist all over our bodies. It is shameful that we still have not agreed on the specific cause of salt-sensitive hypertension, which is so common and thoroughly researched. But with a bit of luck, this age of healthcare practices will soon be seen the way we see Victorian age healthcare practices. We don't know shit about medicine, but we can read, so trust me, I'm a doctor. We'll just need to bleed you out a bit, let some cold wind in to let the bad air out, and let some leeches suck on you. It hasn't changed as much as we'd like to believe. In other words, medicine today is not that far separated from bloodletting. But I will leave you with this message. Like attracts like. Form follows function. Bad medicine is the result of rigid minds, laziness, overconfidence, pride, and scoffing at ideas that are not canonical teachings. And sometimes so out of the box we all have to laugh. Beta amyloid plaques are pretty specific, but if someone told me they found Alzheimer's in feet, of course I would laugh. But I would also investigate with an open mind, while cracking jokes, of course. If you told me you had Alzheimer's in your penis, I'd say there's no way your wife is going to believe that, but I would still keep an open mind and have someone else look at your penis. What if we brought more awareness to these possibilities? When more people see the logic, we get more people on board. Form follows function. Like attracts like. More people bring more money. More money yields more scientists and better science. The street light covers more area. You finally locate your keys and suddenly the world opens up a little more, and medical science is a new animal entirely. What if millions of people finally get a definitive answer to what is ailing them, where it came from, how to treat it, and, as form follows function, paradigm shift becomes paradigm drift as the street light continues to expand. I think that would be lovely.